My name is Lisa Wagar. I'm an assistant professor at the University of California, Irvine. I received the runner-up prize for the Michelson, Philanthropies, and Science Prize for Immunology for my essay on deciphering immune responses to vaccines and infectious diseases using human immune organoids. I've always dreamed of being a scientist ever since I was a little kid. I was very excited about understanding how the world works and taking things apart and putting them back together. I was drawn to the field of immunology because it became more and more clear with my education and my training that immune cells play a crucial role in virtually every human disease. I've been studying the human immune response for over a decade now. My current work is focused on using immune organoids, which are a mini organ in a dish, to try to understand connections between immune cells. So we create these mini organs out of tonsil and other lymphoid tissues. We watch these cells reaggregate in vitro, and then we can understand what the immune response looks like to vaccines and infectious diseases in culture. My future goals are to improve human health through enhancing vaccine design. We hope that the immune organoids will leverage our ability for precision medicine and enhancing the immune response to infectious diseases. The Michelson Philanthropies and Science Prize for Immunology comes at a very crucial time for my research. I started my lab at UC Irvine in July of 2020, and since then, my team is focusing on further enhancing the physiological relevance of immune organoids derived from primary human tissues, and hopefully leveraging that information to improve future vaccine design.
Hi there, my name is Lisa Wagar. I'm an assistant professor in the Department of Physiology and Biophysics here at UC Irvine. Today I'm going to tell you about my work on deciphering immune responses from humans to viruses and vaccines in a tonsil organoid platform. My research interests lie in understanding human immune responses against infectious diseases and vaccines, with the ultimate goal of trying to predict who's going to make a robust, durable, and a protective immune response, and trying to contribute to vaccine and adjuvant designs that will promote protective responses in as many people as possible. One of the major barriers that exist to addressing these types of questions has been a relatively limited set of tools to help us investigate human adaptive immune responses at the site where they're forming. It has been surprisingly difficult to identify correlates of protection, aside from neutralizing antibodies, from peripheral blood studies. Dr. Donna Farber, Dr. Ali Alabedi, and others have recently published beautiful work examining T and B cell responses from human lymphoid tissues. But really, the logistics of doing such studies can be a major barrier to research, and sample sizes often need to be quite small for these types of studies. On the other hand, animal models have really had a limited ability to recapitulate the constellation of factors that contribute to normal human immune variation, including age, sex, immune history, genetics, the environment, as well as many other factors. So rather than trying to eliminate these factors, my work has aimed to embrace the chaos of human diversity and try to study these differences to better understand host factors that account for immune variation. One strategy to overcome these types of issues that I just mentioned is to try to develop a representative in vitro model of the human adaptive immune response that would help us to bridge the gap between preclinical animal models and clinical trials. I focused on using tonsil tissue as a source of material because, all things considered, it's really one of the most accessible lymphoid tissues from otherwise healthy patients. This tissue is typically discarded during tonsillectomy surgeries, of which there are more than 500,000 performed in the US annually. And common indications for surgery include tonsillar hypertrophy, so just enlarged tonsils, and obstructive sleep apnea. In addition, tonsils share many functional similarities with lymph nodes, including germinal centers. I've pointed out some follicles here that you can see from a tissue section, And these germinal centers can lead to affinity maturation of the antibody response, just as you would find in a lymph node. To better study the human adaptive immune response, we developed a novel immune organoid platform. An organoid is an in vitro model that attempts to recapitulate the function, cell composition, and/or the structure of an in vivo tissue. We developed a reaggregation style organoid method that's derived from primary human tonsil tissues with a focus on 3 main points. First, that the cultures would need to adequately mimic the original structure and function of lymphoid tissues. That is, that they're capable of coordinating an antigen-specific B and T cell response. Second, that this platform would enable us to do more mechanistic experiments through, for example, cell depletions or enrichments or other manipulations. And third, that they could be prepared with sufficient throughput and scalability that we would be able to look at hundreds of cultures from the same individual, stimulated with different conditions. Ultimately, this is the workflow that we developed to produce immune organoid cultures. So we receive these tonsil tissues after the patient's surgery. We then mechanically dissociate the tissues down to a single cell suspension, and we can actually cryopreserve the roughly 1 to 6 billion cells that we recover for long-term use. When we're ready to prepare these cultures, we simply thaw the cells and plate them into a non-stick culture device. We found previously that Teflon-treated transwells are especially good for this purpose, and they allow us to maintain the cells at a very high density while still providing sufficient nutrition to the cells through media in the outer well. We can then immediately add our antigen of interest. In my case, I've focused largely on influenza viruses and vaccines, and then we leave the cells to culture for up to 21 days. After a couple of days, the cells will actually start to reaggregate and form defined structures. And you can see from these stereoscope images at the bottom here that the reaggregation is actually macroscopically visible, and the aggregation is much more pronounced when we add an antigen, in this case live attenuated influenza vaccine here on the right, compared to our unstimulated control cultures. We also show that tonsil organoids derived from many donors are capable of responding to influenza vaccines. Here I'm showing you day 7 data, where we measured the frequency of plasmablasts out of total B cells, here on the left. What we found was that with LAIV, live attenuated flu vaccine stimulation, we get a significant increase in plasmablast differentiation compared to the unstimulated control cultures derived from the same patients. And you can also see right next to it here that most donors also respond to the vaccine by secreting influenza-specific IgGs into the culture supernatants. Another benefit of this platform is that we can track responses longitudinally by making replicate cultures or by repeatedly sampling from the same culture supernatant for the presence of flu-specific antibodies. Here I'm showing you just one example donor where we followed the typical trajectory for their response for up to 3 weeks, and we found that flu-specific antibody production started to be detectable around day 7, and it ramps up until around day 14, where it kind of plateaus, as you can see here. We're also able to track antigen-specific T cell responses simultaneously, as shown here on the right, where we looked at CD8 T cell tetramer staining from an HLA-A2 individual using the immunodominant M1 epitope. We found that the CD8 T cell response could expand as much as 10-fold within 7 days. I also wanted to show you an example of how we can use tonsil organoids to perform more mechanistic experiments using human primary samples. So as I showed you before, we made organoid cultures with either the native cell composition of their tonsils, shown here as the no depletion condition, or I depleted total CD4 T cells from the starting pool of tonsil cells on day 0, and then cultured these cells into organoids in the presence of live attenuated flu vaccine for 1 week. After dividing the cohort based on the patient age, I found that organoids derived from our youngest patients, shown here in red, were basically unable to produce a flu-specific antibody response in the absence of CD4 T cells. You can see that these same patients under wild-type conditions were perfectly able to produce flu-specific IgGs. When we looked at the antibodies that were produced from the older kids who were still able to sustain an antibody response when the CD4 T cells were absent, we did find that the quality of these antibodies was lessened. So this finding was well matched with what we know from animal models of germinal center responses, which is that CD4 T cells are critical for providing help to B cells, and they play an important role in shaping selection. I wanted to conclude the data portion of this talk with a big picture overview of what we hope to achieve using this organoid platform in the future. Our goal is to combine information about the host, including donor demographic information, cell composition from their lymphoid tissues, and then a variety of readouts from immune organoid cultures prepared from their lymphoid tissues and stimulated with a variety of different vaccine antigens and immune modulators, and combining that with information from blood-derived measurements to help us build a more predictive model that will help us define the essential features of a protective vaccine response. Ultimately, we hope that these findings will one day contribute to improved vaccine and immunotherapy design. Finally, I would really like to thank my mentors, trainees, and collaborators for their contributions to this work. The organoid model was developed during my postdoc in Mark Davis's lab at Stanford, and I would like to thank him for his continuing guidance and advice. Thank you to my scientific and clinical collaborators at UCI, Stanford, WashU, UVA, and the NIH for reagents, patient consenting and materials collections, and performing assays that were related to this work. Thanks also to our funding sources for supporting this work. And finally, I would like to recognize my wonderful group of trainees, shown here in the picture, and on the left their names are displayed, who bravely joined my lab in the middle of a pandemic, and they're doing exceptional and really creative work. Thank you very much for your attention.